Hey, what's up guys, welcome back. So in this video we're going to be talking about bacterial morphology and the different groups that you can classify bacteria into. Now it is going to be a start to a series of videos that kind of builds its way up to looking at the individual bacteria and what kind of diseases they cause individually. So before all of that, it's really important in microbiology to lay the foundation very well from the get-go, to understand the simple concepts very clearly and logically, so that when you do move on and you do study the bacteria by themselves, you understand why a certain antibiotic doesn't work, or why a certain antibiotic does work, or why when you look under the microscope something looks a specific color and a specific shape whilst other bacteria look different. So all of this is really important, and not to mention, of course, if you understand the individual units of a bacteria, you understand why certain bacteria can cause specific diseases and specific symptoms, whereas other bacteria cause different ones. So that's all really important, and hopefully what I'm going to do is take you through it step by step and logically, so you can understand it very well. Okay, so let's get going. So when in microbiology, the easiest way to look at bacteria, or the first stage in your mind, is to picture bacteria in three different groups. Okay, three different groups. So bacteria are either gram-positive, gram-negative, or they're put into a different group, because there's always exceptions to the rule, right? But just think of it this way, that bacteria are either gram-positive or gram-negative. So at this point now that we've kind of created two imaginary groups in microbiology bacteria, we now are presented with a problem. It says here gram-positive, gram-negative. Okay, so we need to understand now what is gram, like what is a gram stain, and what about a bacteria will make it positive, and what about it will make it negative. And if you're quite observant, you would have noticed that this is purple and this is red. What about it makes the bacteria purple, and what about it makes the bacteria red? So all of this, we're going to take it step by step, and we're going to hopefully understand it. Okay, so the first question in your mind is probably, okay, so what is a gram stain and why is it important? So there are several things that we need to do to understand this, okay? And we're going to take it step by step. Rather than me just info dumping on you, you can just take it, you can just follow along with me, and hopefully by the end of the video everything will be very clear. So the first thing we're going to do is briefly explain what a gram is, really quickly, because we'll build on our knowledge shortly. The second thing that we're going to do is that we're going to understand the basic structure of a bacterial cell wall, okay? Because this really does, the bacterial cell wall is really what determines if a bacteria is gram-positive or gram-negative, okay? And then after we do that, we're going to then look back at the gram stain, the process of, or it's actually like a test. So we're going to look back at the gram stain, and we're going to talk about it in a lot more detail, so that hopefully you can understand everything. And then after we do that, okay, we're going to talk about the individual components of a cell wall, and I'll tell you why that's important when we get there. And then finally, by number 5, since at this stage the importance of a bacterial cell wall and why the bacterial cell wall shapes bacteria, it creates different shapes of bacteria, why then it's important to study the different classifications of bacteria and how that can help us as doctors. Okay, and then hopefully that's the end of the video, and you would be happy to have learned some significant microbiology. Okay, so all of this will then set us up nicely for future videos, where in the next videos we'll talk about the metabolic characteristics of bacteria. Then after that we can talk about what kind of things bacteria have to harm us, like what weapons do they have to harm us, and what defense mechanisms they have to protect themselves. And then once we kind of built the foundation really nicely, we can then go on and talk about the different classifications of bacteria. And this bit is really complicated. Like if you don't study it well, when you study individual bacterial organisms, you'll be really confused. So we're going to take our time on this one and really make things a lot clearer. Okay, so let's get going. Okay, so here we said we'll do a quick, description of what a gram stain is. So this process here is called a gram stain. I haven't guessed already. A gram stain is a test that we do. Its purpose is essentially to figure out, to differentiate between two types of bacteria. Remember we said at the beginning, bacteria can be classified into two groups. Well, a gram stain, it is used to separate the two groups from each other, and this is done by differentiating the color that comes out of this test. So either the color will be purple, or it will be red. If the bacteria turns out to be red, it will be known as gram negative, and if the bacteria turns out to be purple, it will be known as gram positive. And so what we do is that we kind of put different stains. So pretend that these bacteria were all colorless first. The first stage was to put this purple dye on, and then after that we add this red dye on, and then we add this thing here, and then at the end we're left with either bacteria that are red or purple. That's essentially what a gram stain is, and that's how we separate the two groups of bacteria based on color. Okay, so to begin with, it's really important to understand the bacterial cell wall, like the importance of bacterial cell wall, because it's the bacterial cell wall and how it structures and what's inside it that will ultimately make it either a gram positive bacteria or a gram negative bacteria. So let's just look at the function of a bacterial cell wall first. So there's three main functions really, and it's almost common sense. The first one is that the bacterial cell wall will determine the shape of the bacteria. So later we'll see, like sometimes you get circular bacteria, sometimes you get rod-shaped bacteria, sometimes you get comma-shaped bacteria like Vibrio cholera. Sometimes you get spiral-shaped bacteria. They're all determined by the bacterial cell wall, and how the bacterial cell wall is structured. The second function is protection, of course. This can be protection from anything, from outside organisms, from some kind of trauma to them. And then the third function is transport of things in and out of the bacteria. So if a bacteria is gram positive or gram negative, it's dependent on really two things: the cell membranes, how many cell membranes the bacteria has, and also the thickness So if we look here, this is briefly speaking, so if a bacteria has one cell membrane, it will be known as gram positive. And if a bacteria has two cell membranes, so one inner cell membrane and 1. This bacteria will be known as a gram-negative bacteria. So let me just repeat that. So a gram-positive bacteria has one cell membrane, one inner cell membrane, and then surrounding it is this thick bacterial cell wall. A gram-negative bacteria has an inner cell membrane and inside, in between them, lies a thin bacteria. So this is kind of a zoomed in picture. So for a gram-negative, okay, so if we go back, that's this one here, the two cell membranes. For a gram-negative, it has an inner cell membrane, okay, which is this. It has an outer cell membrane, which is here. And then in between that, you have a cell that is very thin. This is the characteristics of the bacterial cell wall of a bacteria that's gram-negative. In contrast, here for gram-positive, it has one inner cell membrane, and on top of that, surrounding it, is a very thick wall, cell wall. Okay. So let me just repeat that. Maybe there's some of you that really know this already and just want me to move on, but it's really important to emphasize. So it's the structure of the cell wall that a bacteria has that will tell us if that it is a gram-negative bacteria or it's a gram-positive bacteria, right? Okay. So before we move on, let's just understand, okay, why are we studying this? Like, why am I telling you if something's gram-positive, something's gram-negative? Why are we talking about bacterial cell walls in so much detail? Why don't can't we just skip this and just go on to the actual organisms? And the answer is that we need to know which are gram-positive and which are gram-negative, because this can tell us what kind of antibiotics work on different bacteria. For example, certain antibiotics will not work on gram-negative bacteria because of this outer membrane, whereas those antibiotics can work easily on a gram-positive bacteria because it can quickly go through because there's no outer membrane on a gram-positive bacteria. Also, it's really important to understand the cell wall because we said before that it determines the shape of different bacteria. So if you're looking under a microscope and you see a circular type of bacteria, then that might indicate something like Staphylococcus. Whereas if you see something that's more rod-like or Bacillus, then that can tell us, oh, this might be Strep, for example. Okay, or if it's spiral in shape, it can tell us, oh, okay, this might be syphilis. And then the final reason really is that there are specific things in a bacterial cell wall that are used by the bacteria to harm us, and it can cause certain symptoms, so it's important that we study them. And also, those specific things on a bacterial cell wall can also be used by our immune system to target the bacteria, so we should be aware of those things. And in a laboratory, it can help us to identify different bacteria because of the components of the bacterial cell wall. Okay, so let's go back to gram-positive and gram-negative. So there are two types. So there's gram-positive and then there's gram-negative. Okay, and here is the same image for your reference. So let's go over it quickly. So gram-positive bacteria have 2 layers. So here, this is gram-positive right here, okay? So it has your cell membrane, that's your first layer, and then the second layer is your cell wall, right? Now, it has and in contrast, a gram-negative has 3 layers. So it has, number 1, your inner cell membrane, then it has this thin cell wall, and then it has your outer cell membrane. Okay, a gram-positive has a thick cell wall, okay, whereas the gram-negative tends to have a very thin cell wall. Okay, and as we said, a gram-positive has 1 inner membrane, whereas a gram-negative bacteria will have 1 outer membrane as well as an inner membrane, right? Now, these are extra features here, okay? A gram-positive will not create endotoxins. So endotoxins are essentially, let's call them weapons that the bacteria have to harm us, to harm the host. So gram-positives, they don't have endotoxins, whereas gram-negatives, they have. And the way that I remember this is that endotoxins, N for negative, okay? That's how I remember it. Whereas there's no N in positive. Okay, let's go back. Okay, both of them, gram-positive and gram-negative, create exotoxins. So again, these are weapons, but different types of weapons. We'll go into detail of them in the next video. Okay, the way that I remember that both of them have exotoxins is that it's X. I remember it as 2 lines. So both. That's just the way that I remember it. Okay. Gram-positives have these things in their cell wall called teichoic acids that can be very, very useful for us to identify different gram-positive bacteria. Whereas gram-negative bacteria have these things called porins that can help these are porins here. It can help transfer chemicals closer to the inner membrane of the bacteria, whereas gram-positive, they don't have this, and that's mainly because they don't have an outer cell membrane, so they don't need porins. Okay, and gram-negatives also have murin proteins, and these are essentially proteins that bind the inner cell membrane to the bacterial, to the cell wall here. Okay, so these are, for our discussion here, these 3 things are really the most important. Okay, if you do want to add something in from now, this will be important, the endotoxins. Okay, so here is a brief recap. You can kind of go over this by yourself, or just use it, take your time to understand it and absorb all the information in. All the labels are there, and just to kind of clarify, the endotoxin is actually called the lipopolysaccharide, the LPS. Okay, lipopolysaccharide, and as you can see here, it's located on the outside. And again, it's like a weapon that the bacteria has. Okay. Okay, so here we said that there's really 4 main components of a bacterial cell wall that you should understand. Number 1 is the cell wall. What's it made of? Number 2 is this LPS here, the endotoxin. Number 3 is the porin. Okay, and then number 4 is the teichoic acids. Okay, so peptidoglycan is essentially these purple, and they're actually sugars. Okay, they're sugar molecules that will make up the cell wall, and they're kind of stuck together, and they're arranged in a mesh-like structure. So you can kind of see it here. It's like they're kind of in a mesh shape between it, and the space in between it is quite important for the test, for the gram stain test that we're going to talk about. The cell membrane, of course, that's an important structure of the bacteria. Remember that gram positives have one inner membrane and gram negatives have two membranes, so one inner and one outer. Okay. Thirdly, you have your lipopolysaccharide, which is your endotoxin. So remember, this here is your endotoxin. It's located, it's found in gram bacteria, and it's used as a weapon against the host. Porins are used by gram-negative bacteria to transport molecules past the outer membrane towards the bacteria, because remember that the membranes are they're lipid soluble, so they won't let molecules go through it. It's just physiology, isn't it? That's why there's kind of channels here that will allow molecules to come closer to the bacteria past this barrier. Okay, and then gram positives, they have teichoic acids, and these act as antigenic markers. So if we put it on the diagram, we might say that this is a teichoic acid here. Okay, and maybe when we are in the lab and we're trying to test for a certain bacteria with a certain teichoic acids, let's call this teichoic acids Ta1. Okay, let's call it that. So then the person in the lab, he has a specific test that can identify teichoic acid 1, and then if he finds it, then he'll be like, oh, then it's this type of bacteria. Okay, that's the purpose of teichoic acids on gram positives. Okay, so this was basically to just give you an understanding of the components of the bacterial cell wall. Okay, so let's look at gram staining. So what is gram staining? So now that we understand the difference between a gram-positive bacterial cell wall and a gram-negative bacterial cell wall, let's understand what the stain is and how you get a purple color or a red color. Okay, so as we said, that bacterial cell walls are composed of different peptidoglycans. So these are sugars, and that's what makes this mesh-like structure. Okay, it helps us in gram stain classification. So essentially, it's a test, and we use several dyes, just like we briefly saw before, like a purple one, a red dye, and a colorless dye. We use this to tell us what kind of bacterial cell wall the bacteria has. So does it have a thick cell wall with one membrane, or does it have two membranes? That's essentially the purpose of a gram stain. So we can pretend here that this is the first plate, and you have bacteria that you do is that you add crystal violet, okay, the purple dye, to this colorless plate of bacteria, and then you dye it. Okay, and then the second step, okay, is that you add iodine, okay, onto the bacteria, and then once you've done that, you wash off the dye using alcohol, and what that will do is that it will get rid of any kind of excess dye. So the dye that's absorbed is going to remain. So you can see here that the round are dyed purple. So in other words, these are gram-positive bacteria. Okay, whereas the rod-shaped bacteria, they didn't absorb any dye, and that indicates that they're gram-negative. Okay, but the point that I want to make here is the importance of the bacterial cell wall. So let's look at this. Okay, so this in the laboratory, this was the cell wall was the rod-shaped bacteria. Okay, so what happened is that when we were in the first stage here, okay, we added the purple dye here. We added crystal violet. Okay, and what happened is that the crystal violet came and it was absorbed in between all of the sugar here. Okay, to the sugar, look, you can see here, look, there's a mesh. Okay, it's almost like a net, so for the dye to absorb inside the sugar cell wall. Okay, but the problem with the gram-negative, which is why it wasn't stained at all, is because there's a lipid-soluble membrane here. The membrane blocked it, so it couldn't get absorbed by the bacterial cell wall here. So instead, it was easily washed off. Remember in this stage here, everything was covered. In this stage here, everything was covered in dye because we've kind of flooded the agar plate with dye. But then when we went to wash it to see what bacteria actually absorbed the dye and which did not, we found that the bacteria that had an outer cell membrane didn't absorb anything. Okay, it was only the bacteria that had only an inner membrane. It didn't have an outer membrane, so the iodine was absorbed, and that's why these were stained and these did not stain at all. And so we don't just stop there. What we want to do is that we want to stain these ones because we want to see them on the microscope. So we add safranin here, and what that does is that it can actually penetrate the outer cell membrane, and it will stain those bacteria red. Okay, so that's why here essentially gram-negative bacteria are red and gram-positive bacteria are purple. Okay, so I hope that makes sense. Okay. Okay, so now hopefully it makes sense that gram-negative bacteria are red and gram-positive bacteria will be purple in a gram stain, and that helps us to split the two different bacteria into different classifications. Okay, so now let's move on to the next stage, and the next stage is, well, since we understand that the bacterial cell wall is really important in forming different shapes of bacteria, it makes sense that we should understand all of the different groups of bacteria according to their shapes. Because if we're looking under a microscope and we're looking, for example, for let's say Streptococcus, okay, we need to understand that Streptococcus belongs to the group of bacteria that are shaped like rods. Or if we're looking for Staphylococcus, we need to understand that Staphylococcus belongs to a group of bacteria that looks like cocci, balls, circular shaped. Okay, so essentially, what are the different shapes that bacteria can come in? So there are three shapes really, like three main shapes. It can either be circular, it can either be rod-like, or it can either be spiral in shape, a spirochete. Okay, there are others, but these are the main ones that you should burn into your brain. Okay, so a bacterial cell wall can be structured in a way so that the bacteria will either be spherical in shape, rod-like, okay? And the reason why I have this here is because I didn't want to confuse you. Now I'm going to remove it, okay? And I'm going to show you what I mean. So things just got a lot more complicated. Not really, actually. So we said that the bacterial cell wall can structure the bacteria into either a sphere, a rod, or a spiral, right? Okay. Now, depending on the type of bacterial pathogen, okay, it might be Klebsiella, it might be Neisseria gonorrhea, it might be Staphylococcus, it might be Clostridium difficile. They all will be composed of, they'll all kind of be grouped differently, okay, or they'll look different, but the basic structure will either be a sphere, a rod, or a spirochete. So some bacterial infections, when you look under the microscope, they're in pairs, okay, like Neisseria gonorrhea, for example. When you look at them, they're in pairs, okay, and they're spherical. Other ones are in chains, okay. So, for example, some enterococci, they're in chains, and they're spherical. Other bacteria, like Staphylococcus, they're spherical, but they're clumped together, okay. Now, some bacteria are rod-like, but they're rod-like and they're in pairs, okay. Some are in chains, like Streptococcus, and some of them have tails, okay. So all of these things are important for us to understand as doctors, because different infections are caused by different microorganisms that have different shapes. And then finally, you can get spirochetes, and spirochetes are either Borrelia, which is the cause for Lyme disease, Treponema pallidum, which is the cause for syphilis, or you can get other kind of spirochete organisms. Okay, so this is a more graphic image. So here you have your cocci. They're in pairs, rod-like bacteria. Okay, these are individual and these are in pairs. Okay, and then here you have your spirochetes. Okay, so essentially that's what you should understand. So just to go back and recap, bacteria, the most basic units of bacteria are either spherical, rod-like, or spirochete, and these are determined by the bacterial cell wall. And different infections have organisms that are grouped differently. Maybe they're in pairs, maybe they're in chains, or maybe they're clustered together. Okay, so there are a few exceptions. So Mycobacterium, which is like tuberculosis, this does not stain using a gram stain. It's we use acid-fast staining. Spirochetes here, which are these tend to be so small that it's really hard to stain them. So instead, what we do is we use fluorescent dark staining. And mycoplasma, this is an exception to the rule because it doesn't even have a cell wall at all. Okay. Okay, so that's the end of the video. I hope this was useful. We're going to do a quick recap now, but the main point of the video was to get you to understand what is a gram stain. So a gram stain, or you can pause the video and try and answer it for yourself. So a gram stain essentially is a test that we do using different dyes to identify if a certain bacteria is gram positive, i.e. purple, or gram negative, i.e. red. And the reason why that's so important is because we said from the beginning, didn't we, that the best way to look at microbiology bacteria is to split bacteria into two groups. They're either gram positive or they're gram negative. So the second thing that we talked about was what's the difference between gram positive and gram negative cell walls. So let's quickly just go back and talk about that. Okay, so we said that the main thing in a gram negative is that there's two membranes, okay, and there's a thin cell wall, whereas in gram positive there's one membrane and the cell wall is very thick. And we said the reason why this is so important, okay, the reason why it's important to understand the differences is that some antibiotics won't work on gram negative bacteria because of the outer membrane blocking it, whereas antibiotics tend to work easily on gram positive bacteria if there's no resistance. And then we said that there's different components of a bacterial cell wall that help the bacteria in functioning, especially the lipopolysaccharide, which is an endotoxin, and that's used as a weapon by the bacteria against the host. Okay, and then finally we said that since we understood that the bacterial cell wall is responsible for creating the shape of the bacteria, we should understand the different morphologies of bacteria so we can identify them under the microscope. So remember, we said that there's three different basic units of bacteria. Either it's spherical or it's rod. There are also other types, but let's just focus on these three. Okay, so in the next video what we're going to do is that we're going to proceed on to understanding the different metabolic characteristics of bacteria. So there's things called facultative anaerobes, there's things that some bacteria are spore forming, some bacteria are not, there's other bacteria that are known as siderophores that use they use iron. Some bacteria, they're intracellular organisms, whereas others are extracellular. So all of these, we're going to understand them so that in the after that, the third video, we'll go on to talk about what are the weapons and defense mechanisms that bacteria use when they're invading the host. And then once we create a strong foundation, we'll then go on and talk about exactly the different types of classifications that bacteria are split into, so that everything is perfectly organized in your mind, so that when you do come across a name of a bacteria, you'll say, oh, okay, so this bacteria is gram positive and it's a spore forming bacteria, and it's responsible for this disease because of this. Everything will be very, very well laid out in your mind. Okay, so thank you very much for watching. I hope you reached the end of the video and I hope that you found everything in it useful. Please do like, subscribe and share if you found this video useful, and I will see you in the next video. Thanks a lot.